Hi, my name is Yiqian Zhang. I'm from Waseda University in Japan. Today, I'd like to introduce our research on RNA multiplication m6A. This research is about the identification of m6A-associated RNA binding proteins using an integrative computational framework. RNA multiplication is biochemical modifications of RNAs that have functional regulations without a change in the RNA sequence. There are several kinds of RNA modifications in mRNA. Among them, m6A is one of the most common and abundant one. m6A is preferentially located near 3' UTR, so it has influence on some essential RNA activities like translation and decay. About m6A regulators. m6A regulators are RNA binding proteins related to the formation and function of m6A. There are three kinds of m6A regulators. They are writer, eraser, and reader. This figure shows the dynamic process of m6A and its some well-known regulators. Interaction between m6A and m6A regulators plays important roles in essential RNA activities, and it is also closely related to cancer progression like AML. For example, as this figure shows, m6A writer MELT3 is necessary for maintenance of the leukemic state. So studying m6A and its regulators enables us to develop a better understanding of gene regulation mechanism and leads to potential therapeutic opportunities. In this study, we collected myriad data from several independent studies and RBP's binding data, and then we designed an integrative computational framework to identify m6A regulators. In this framework, we first identified reproducible m6A regions. Then, using the RBP's binding data, we performed the enrichment analysis and built a random forest classifier. For the enrichment analysis, we used a permutation test. We obtained control regions by sampling 1,000 times from the rest of regions in genes with m6A. For each RBP, we calculate an enrichment ratio by this equation. Then we calculate p-value and performed multiple tests. We identify some non-m6A regulators as these green arrows point. We found that CPSF6 is common in both human and mouse. In addition, we identified a potential m6A regulator RBM3 in the mouse cell line. RBM3 is an important regulator for circadian gene expression by controlling APA, so we speculated that RBM3 could be a potential m6A reader that regulates the APA by interacting with m6A. Next, we built a random forest classifier based on RBP's binding data. And this classifier achieved the competitive performance compared with two sequence-based ones. We also plot the feature importance to check RBP's contribution. Apart from non-readers and writers, we noticed a RBP named LFL1. LFL1 has been reported to be a m6A repelled RBP. These results suggest that our framework helped to infer interaction between m6A and m6A regulators beyond the sequence level. In conclusion, we designed an integrative computational framework for the identification of m6A-associated RBPs from reproducible m6A regions. Our framework is able to identify non-m6A regulators and also found some potential regulators such as RBM3 for mouse. And our framework also helps infer the interaction between m6A and m6A regulators, like actions of reading and repelling beyond sequence level. Thank you for listening.